Good morning, all. I welcome you all to this 22nd day of our CSIR online training. Today, in this class, we are going to discuss about the topic evolutionary behavior. Before I enter into the section, please confirm am I audible? Uh, Good morning. Please let me know am I audible? Can you see my slides? Type it in chat box. Yeah. Thank you for your response. Now we are entered into the session. Okay. Yesterday we discussed about the session 2. We divided into 3 sections. In this session 2, we discussed a lot about the 2-mark questions, very important 2-mark questions part. In that way, we are going to discuss today. We are going to discuss about the topic evolution and their behaviors. Okay, in this topic, many of the 2-mark questions are there in CSIR examination. In today, I am little bit concised because we can't cover the whole unit, right? So important topics I am included. I will upload the another video in evening or tomorrow. I will upload the remaining important parts. Okay, in this class I am included the very important topics about the unit 11, that is evolution and behaviors, and some more topics are available in the unit 11. I will cover. I try to cover in the another video. I will upload as soon as possible because in this 1 hour we can't finish the whole unit. Okay, now we are entered into the session. Okay, first topic is the our Lamarck Theory. All of them know. We are all studied in a 12th and 11th standard. In this Lamarck Theory, what they said, okay, that is a development of the neck, long neck in the giraffes. How it is developed? Why it is developed? It's developed for the need of food. So this Lamarck theory explains, okay, explained by the in that 2-mark way. Who explained the theory? Which what is the theme of theory? These are all the things are in the RCSI examination. So I am included this content. Are Jean Baptiste Lamarck. Okay, Jean Baptiste Lamarck. He explained the theory of acquired characteristics. This is a theme. In this theme itself, he explained the original neck size and its how it is changed. Okay, using the in that period, he measure the measure the normal neck and the giraffes, the long neck. In that normal neck, that in that generation, that is 18,800 year, he's seen the both generations. Some of them in this height, some of them in this height, long and short. So he okay believe this is the evolutionary change. Is the generation size somewhat generation oldest generation is the young generation, so for their food need it's grown, it's developed their neck. Okay, he discussed about many animals shape and their forms. Okay, many of the many discussions are discussed by the are this Lamarck. So only this theory is called as Lamarck. It's the theory about the acquired character, that is depends on the shape and their form. Form means that a long, short, or activity, behaviorals. Okay, everything. The changes is they are used and use and disuse. Use and disuse means it may be good, it may be not usable. Okay, some in their evolutionary period, if any portion is developed in or in animal or anything, that is not needed for their purpose, but that is a disuse of heritable. Okay, in this that point of view, here how's this horse like animal evolved into the giraffes, how it is possible. This is the major theme of the Lamarck theory. And I here also explained many animals evolutions and many plant how it has changed their characters. These are all the things done by the Lamarck. Okay, this is the Lamarckism. In this, some more extra points, because this is, I told this is a 2 mark point of view. In that 2 mark point of view, they may ask any way. So only I am included here. This Lamarck idea of the ideas of evaluation. This Lamarck idea of evaluation is uh, this is the first theory. Okay, this is the first theory of evaluation. We all know the Darwin theory is the famous one. So before that Darwin's theory, this Lamarck's ideas of evaluations comes out. The order of evolutionary publications also can be asked, can be comes in the is 2 mark of CSIR examination. And another thing of this Darwin's this Lamarckian theory is how it is abundant, how it was abundant. Okay, why? Because the after the Darwin's publication and inheritance of explained thought through the genetics, genetics. Two things are there. First one is the Darwin's publication, then the inheritance or related genetic related theory. Both studies, after this both studies, they accept the Lamarckism theory. Okay, then here they ask the questions for their related to the Lamarckism. And what is the Darwin's theory? Here is a 5 it's some principles are here and some variables are here. How the Darwins explain the theory of evaluation through the natural selection. Okay, natural selection. What is natural selection? It's a the process of differential survival and reproduction of individual variant. It's already exist in the group. This is the natural based on in this what they mention differential surveillance. The process of differential surveillance means that can be surveyed in different for various generation. It can be surveyed in the various conditions. And individual variant, that reproduction of individual variant means that become this one in that group, that is the make a individual variance. These two things are very important: surveillance, differential surveillance, and reproduction of individual variant. Okay, in this we that having the different principles, right? This is one point of view, because many of them is divided into different types. Then this one of the some of them are divided into three types. Some of them are included the heritable variations. One in this principle of variations. What is principle of variations? Among the individuals within any population, any individual in any population, there is a variation in the morphology, physiology, and behavior. This is the principle of variation. This how it is related to Darwin's Lamarck. Okay, Lamarck said some of them having long neck for their food, to achieve their food. In that group, the needed animal alone changed. That is the shape. That is the variation. So in this Darwin's theory, principle of variations are explained by the they explain individual within any population. There is a variation in the morphology, physiology, and the behavior. The principle of heredity. Heredity in this offspring resembles. Offspring resembles means that is the child, that is the next generation of the first generation, that their parents more than their resembles unrelated individuals. Okay, some of them don't have the similar property, similar look to their parents. In this theory, Darwin explains the principle of heredity and the principle of selection. Some forms of are more successfully at the surviving and reproducing other than the forms in any given environment. In one environment, many of the forms, forms means that is the group are successfully surviving in the pre and reproduction, that producing the reproduction. Because in this selection, why they mention the selection? So in this group they can adapt to, that is survive, differential survival, that can be survived in the particular environment, and it can be reproduced at the particular it can be reproduced at the environment. This is the two different point of view of the Darwin's. First is depends on the variation and heredity and selection. Okay, this is another explanation. Individuals variation and offspring resembles variation, and the it's a surveillance based on the successful surveillance. And hereditary variation means it's the heritable difference between population over time. That particular time itself it vary in the population. Okay, this some basics of the Darwinisms. Okay, they explain depends on evolution, common descent, and species multiply, how it is multiply, and groundalism and natural selection against Lyell's and against Lamarck. The Lyell's say this Lyell's means she is one theory. He is also explained on theory. In that theory, it's about sequence, species sequence. Here they mention the Lyell is explained the theory of species sequence. It's a 2 mark based on this. They are asked theory theme and the author. That is the theory explained per scientist name. So theory of accurate characteristics is a Lamarck. Theory of the sequence, species sequence is explained by Lyell. Okay. Here these two theories are uh, or it's defense by the Darwin's theory. These two things are against to the Darwin's are against to these two theories. Okay, here what's in their said what they said, species are not stable, but in that Darwin's theory the species are stable. It's perfectly not perfectly adapt to the environment, and species are quickly created. These things are opposed by the Darwin's theory, and individual changes are inherited, not inherited. Here this is also opposed by the Darwin's theory. Okay, next is the adaptation. How it is adapt to the environment, why it need to adapt. That for the surveillance it's need constant struggles. Okay, the thing is constant struggle. Okay, there is a need of water, there is a need of food, any foods, everything. It's a constant struggle within the ecosystem. It's very very important in the exam point of view, because how it is developed, the adaptation how it is developed. If there is a struggle, once a struggle, it's went na, they won't adapt to the environment. That they daily every day that face many different struggles. But in this way, in this adaptation, how it is developed means constant struggle within the ecosystem. It's very important. Okay, how adaptation, what is the possibility? Any characteristics can help to survive the at the environment. It's like here they explain with the camel. You all know about this concept. Camels store the larger amount of water in their stomach. How it is developed? Okay, that needs water. That is that's moving through long way of the desert area. So in that area, that needs the heat. That needs the water. There is no water, so it's stored in the stomach of the its stomach. It's one thing, and it's backbone development. This is also for their heat resistance. Okay, this is developed. This all things are, and their neck bend. This neck bend also it's developed for their movement as means in the desert they face the deep air flow. In the desert they face the deep air flow. To face the air flow, this camels are having that neck movement. Those are all the evaluations and the adaptations of the that camels. Then organism is successful adaptation is more likely to survive other than the other elements, the other organisms. So adaptation is very very important in the evaluations. Here's some more explanation. What is the adaptation? That's an example. Here they explain with the that life experiments. And an adaptation, an inherited characteristics that increase an organism stands change for the survival. Okay, inherited means that is a developed in the genetic. It's that group of animals that change their internal characters. Then adaptations is vary for the population. If there is a small population, the adaptations differ. If there is a large population, that adaptation is different. It's take thousands of years, and it make a difference in the DNA level. Okay, that is very important thing. This adaptation make a changes in the DNA level after the over period. Okay, this is the struggles. What are the possible struggles for animals? What are the struggles for the plants? Because based on the struggles alone, they develop their adaptation. Animals are struggles, or what is struggles? It may struggle. They are based on the uh, food, based on the water, for protection against the weather, and that is the camel concept, and their external skin skin hair, that hair development over the skin, that are all the based for the protection against weather. The bears develop their fur on the skin, that is all very very important for the protection of weather and against the being eaten by predators. Some this anteater having that sharp um, edges on the their whole surface, that are the eaten by productive and be against being the eaten by predator, and it's against some diseases. Okay, and against accidents means it's movement to safe movement, for their safe movement. These are all the some struggles. And for water, they need a water. For that water production, that is the dryland species called that crotons related, that store the water. And the light, for the taken to receiving the light, that design they develop their adaptation to obtain minerals in the soil, against to the weather, against to disease, against being eaten. Yeah, this is the some of the you all may know that some plants are having the fight that chemicals against the some animals. Okay, even aadatoda, you may hear the word, that is a major food source for that is eaten by many animals, but that is developed their chemicals against the some animals, so that are not able to eat in the leaf, that if that leaf has been eaten by the animal, that has become dead. So these are some struggles. For the struggles, the plants are developed their parts and height, for their shape, change their shape. These are all the struggles and struggles of animals and plants. And how this it's a better, it's for some output, why it's how due to this adaptation, what happened. It's a better copying extreme of it's copying with the extreme of weather, and competing for food, it's escaping from predator. It can be it okay, and the final thing is very important here, survive their habitat, and it's produce the offspring. In this offspring itself, that character become transfer. They 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 also if the first two or three generation, that having that teach to their next generation, after that particular period, that our DNA get difference, that DNA get modified, that is genetically included to the offsprings. Okay, it's a spontaneous mutation. This is the sequence of the after. It's a very sequential step of the adaptation. In this spontaneous mutation, what happened? It's during, it may happen at a S phase, G1 and G2 phases of cell cycle, by the movement of the transposons. Okay, these are all the things are very important, because in this mutation, how it has happened, this kind of questions are they ask. Spontaneous mutation rate in eukaryote is between 10 power minus 4 to 10 power minus 6 per gene per generation. And in this, it's in eukaryote, there's a prokaryotic, that is a bacteria or phages, It's 10 power minus 5 to 10 power minus 7 genes per generation. This is a very important thing. For if there is a eukaryotic, there is a chance of mutation. Spontaneous mutation is a 10 power minus 4 to 10 power minus 6. If there is a prokaryotic or a bacteria or a phage, it may be it's in a minus 10 power minus 5 to 10 power minus 7 gene per generation. And another condition here is for example, Drosophila, it's a male and females are the same strains having the similar mutation rate. So in that it's after the genetic constitutions. Okay, so there is no changes in the male and female. In the all organisms, they are having the difference between the two strains, males and females. It's not having the that kind of thing. Then flies of different strains. However, many different mutations rate, and but they it's some flies are there in this different strain that having the different mutation rate. This is a genetic constituents of organisms affects the mutation rate. So that depends on the mutation rates. It's very depends on the organism. Then many spontaneous errors are corrected by the cellular repair system. That's why only it's the spontaneous mutation happen at the S phase, G1 phase, G2 phase, any cycles of in cell cycle. It's some point of cell cycle. So in that way, that will affect the generation, the that mutations affects the mutations. Okay, this is the various way of mutations. In this way, it's heritable changes in the DNA, and they are essential for the genetic study, and it's make many biological effects. Okay, this the two things we want to. It's a heritable change, and it's essential to study the genetics. It's and its cause, and it makes a biological effects. In this first image, you can see that there first I would like to tell what is a somatic mutation and germline mutation. Somatic mutation occur in non-reproductive cells. A very important thing. Somatic mutations are not it's occur in the non-reproductive cells. Germline mutation occur in the cells that gives rise to gametes. You may know somatic cells means this is non-reproductive cells. Second non-reproductive cell, germline mutation is occur in the given rise to gametes. Okay, first step you can see that mutation occur in the non-reproductive cell. Here the another option is a reproductive cell. After this mutation, the genetic base are modified. This modification of genetic pairs is make a changes in the their population of mutated cell, that make a changes in the population. And some time that carries the it's not carries the DNA, that not make the mutation. There is no changes in the DNA, that will not make a mutation. Then it's another example how it is the it's happen here. The one mutation, the spontaneous mutation is known as the tomatomerism. That tomatomerism is changed by the repositioning of hydrogen bond. Okay, the DNA, that is the tomatomerism. In this, they mention the repositioning of hydrogen atoms in the hydrogen bonding atoms and altering the hydrogen bonding, that makes the incorrect base pair during the replication. So it's very important in the spontaneous mutation, because in the every step they mentioned, right, that will alter the heritable change in the DNA. At the DNA level, how it has happened, that the DNA hydrogen atoms changing and hydrogen bonding pattern, it's altered. Due to this alteration, the replication is it's confused. In this process, the ability of molecule to exist in the other than one chemical from is called as the chromaterism. The very important thing, the ability of the molecule to exist in more than one chemical form. Here you can understand, here this A molecule, this is adenine, it's having the shift, it's alter the it's alter the base. Here this is a shift, in this shift itself that will change. So here you can see, is it possible? A and C is possible? No. Here, but the chromaterism theory, there is a mutation happens. This mutation makes a bond between A and C. Then all the 4 common base pairs of this DNA have unusual chromateric form. This adenine guanine, it is made this adenine, normal adenine, but that is so in the immunoform. In this immunoform, that is some alteration in the chemical structure. That changeable chemical structure is called as the chromaterism. That one ability of a molecule to exist in more than one chemical form. It's that is so that nucleotides A, T, G, C, that are all converted into the immunoform. That's immunoform. It's helpful for the this spontaneous mutation. Okay, next we are very important, very interesting topic. It's a Mendelism. What is Mendelism? In this Mendelism, here it's we all know this Mendel is known as the father of genetics. He did experiment with the pies, the Pisum sativum. Using this plant, he explained the theory, Mendelism theory. How he explained the theory means with the production. Without this Mendel theory, without this Mendelian inheritance question, there is no CSIR examination question. So very important question. In any point of it, they may ask in the 4-mark question, they may ask in the 2-mark question. It's a very important topic. It is Mendelian theory, Mendelian inheritance. Okay, here this is a parent. This is the parental species. This is a pure breeding of yellow, pure breeding of green. Then first generation they got the yellow, but second generation they got the green color. How it is transferred? In this step, in the parent to first generation, that is muted. That is silence mode. So in that F1 generation, there is no green color, but that green responsible for green color pie is silenced in the DNA level. This thing is very important. The F1 F2 generation development. Here another one simple example: pure male and pure female. That is they mention yellow and green. Here they mention yellow and red, green and wrinkles. Wrinkled means that is a one character. Here they mention the here is a round is a character. Yellow here yellow and green is a color character. In the first step they mention color based alone. First one is the yellow, It's a green, it's a combination, then next generation. But here mentioned two characters. One is a color, one is a shape. Here this yellow colored and wrinkled shape. Another is a green color and yellow color and round shape. Here another one is a green and wrinkled shape. These are all the here this first itself. It's both are dominant. So dominant is a capital letter denotion. Small letter means it's a predominant. Predominance means it's a capital letter. Dominant means it's a small letter. That it's here the small letter and it's capital letter. The first generation of all round yellows are obtained. So it's having the characters with the both female and male. But after the next generation, after the second generation, that receiving the capital OR, small, these are possibilities. Okay, it may both are capital, that is a predominant. This any one character alone is predominant, and here is second thing, the round shape alone predominant. But in that final option there is no predominance, all things are dominant. Okay, natural selection and fitness, it's very important, and another topic is focused by CSIR exam point of view. In this natural selection for fitness, what is a fitness? Fitness means that is the after adaptation, that main ability to survey, that is maintain in the one environment, survey or reproduction. Okay, the high fitness means it's a best adapted. So only I mentioned after the adaptation period, they want to survive or want to stable in one environment, that is called necessary for the strongest. Not necessary for the strongest means that having the adaptation, so there is no need to defend. After adaptation, the characters are inbuilt to you, so no need to defend. So there is no need to strong. Then some behaviors having the larger strong teeth, but some behaviors are having small and weak teeth. Why it's having, these are the animal behaviors. So why it's having means based on their food property, that is the beaver with the strong teeth, it's more likely survive and pass down the gene to offspring. It's a one example with the teeth of the animals. And in this another example, here they mention with the animal is having the length, the longer teeth and stronger teeth. Here the another example for the natural selection for fitness, for their surveillance, they develop their long neck. The taller giraffes are more likely to survive and pass on their gene to offspring also. These are only for their surveillance, they are fit to the nature. May here you are not fit to this job. Why they said you can't develop you are not having the characters based on the that job environment. So likewise the animals, those who have the perfect fitness to the adaptation period, that is a new struggle, that only called as the natural selection, that will become develop their long neck in the giraffes. That is depends on all these things are depends on the inheritance and genes level. These changes are transferred to the next generation also. Okay, that thing is a very important thing of the evolutionary part is a modern synthesis. How it is synthesis? First time they mention, just refresh that Lamarck theory. He watch the animals and take the report, and Darwin's also did the experiments with their survivings. They did Darwin's theory. Based on the Darwin's theory, they did the many visual experiments. That is the animal based experiments, animal based the report taking, that is the reviews. And another thing is the Mendelian theorem. The Mendelian theorem he explained with the pi. But the modern evolutionary synthesis, it's fully based on genetic based one, genetic level studies. This based on the genetic understanding and databases, they develop the one tree that is called as in this itself you can understand some a group of bacteria are there, that fungi, animals, it's a full evolutionary group, but they are mentioned the protozoa, algae, plants, simile models, animals, fungi are in one group. Another group that having the proteobacteria, acetobacteria, that kind of things. How they develop? After some period of time, they extract the DNA of animal and they did resequence. After the sequencing, they compare the sequence in the computer level. So it's a modern, it's evolutionary theory. It's a branches of biology. It's a make of branches. Now we are studying the prokaryotic study, eukaryotic study, bacterial study. All those things are branches. It's very important in the evolutionary theory, evolutionary synthesis. With this are the synthesis. Synthesis means it's produced. So it may induce the changes in the genetic level. Okay, what is the normal Darwin's evaluation? What is the modern synthesis? What is extended evolutionary synthesis? In the evolutionary synthesis, Darwin explained the Darwin's replication. In the replication that developed by that replication based study is developed and experimentally did by the Mendelian theorem, this Mendelian inheritance. This Mendelian inheritance is extensively studied by the latest researchers, that is the inclusion of modes of heritability. That is the here one example they provided, the horizontal gene transfer experiment and epigenetics and culturing. These are all the things in the extended evolutionary synthesis of Mendelian inheritance. This is a very important. They may ask in match the followings. The basic theory is replication, that's heredity, thus explained by the modern Mendelian inheritance. This Mendelian inheritance is extensively studied in the modern researchers. Is for example horizontal gene transfer theory, epigenetics and culture. Then here horizontal means after the time period the one character is continuously passed to all this evolution, so that are all these offsprings. So that is kind of then the Darwin's evolution theory about gene, protein, phenotype. This concept, genes are responsible for production of protein, proteins are responsible for the phenotype selection. This is developed and discussed with the concept as a developmental schedules. These are all extensively studied in the topic evolution of developmental regulation. Then variations means that is the that variables, that mutagenesis is rare by SAP effector. SAP effector is a very important concept in the mutagenesis. In this modern synthesis, they explain with the cosmic rays, because cosmic rays are not not make a mutagenesis. It's a rare one. It may cause, but it is a rare one. In the extended period, they explain that the mutagenesis is common as the result of mutation repair is failure. Okay, in this concept, I want to let you know one sometime the mutations is usual one. Okay, in that usual one, that molecules are repaired by our own cell system, but sometimes it's not possible in the nature, so only they mention here is a it's a common, the result of repair is failure, sometimes it's failure. So it's make a some offspring generation gap. Then selection, that's natural selection of individuals. It's studied by the modern synthesis, and the evolutionary, the extended synthesis is helps to mutualism and symbiosis process, selection of on the multiple scales, niche construction involvement, self selection. These are all the things are studied in the modern evolutionary studies based on the selection. The mutualism and symbiosis is a very important concept in the study of extended evolutionary synthesis. Okay, next we want to discuss the concept called aerobic and anaerobic metabolism. You may know about this concept, but once again I want to refresh this concept. Okay, this two thing is very important, aerobic and anaerobic. In aerobic metabolism, the pyruvate is converted into the lactate. Okay, this happen and oxygen to reoxidize the NADH to NAD. This is the electron transport chain process. In this anaerobic process, electrons transport chain involved in the reaction. But in the aerobic metabolism, that pyruvate converted into acetyl CoA. After this acetyl CoA conversion, that is acetyl CoA utilized by citric acid cycle. Citric acid cycle produced the CO2. That this aerobic level, the aerobic happens with the citric acid cycle. Another thing is glycolysis. It's a sequential process of the aerobic condition. It's producing, using the glucose, it's produce a pyruvate. Okay, this is the thing. It's about aerobic and anaerobic conditions. Here, what is the reactants in the anaerobic? Here, anaerobic, the reactant is a glucose. That incomplete process. Okay, consumption is incomplete. So low energy is yield is very low in anaerobic condition. Yield is very low. That is a 2 ATP production allowed. This is a very important point of exam point of view. The high energy is a low energy production happen in the anaerobic. This product is in animal it's a lactic acid product. In a yeast is ethanol and CO2. Here I want to let you know one thing. In an anaerobic condition, that itself they produce the ethanol, that yeast produce the ethanol in that alcohol production. It's production of alcohol and CO2 in the condition, that anaerobic conditions. That the yeast is producing baking, it's all things that happen with the anaerobic conditions. Location, it's a cytoplasm, and stages is glycolysis fermentation. The only thing is the glycolysis fermentation. It's a make a citric acid cycle involved in the step. Okay, next is aerobic conversion. In this aerobic process, oxygen and the glucose is the input. In this input, that having produced is a complete process. After the completion, produce a high ATP molecule, and it's produced CO2 and H2O. Its output, it's happen in the cytoplasm and mitochondria. It's a aerobic process happen in the cytoplasm and mitochondria. Then aerobic process, it's this glycolysis, link reaction, Krebs cycle, electrons transport chain. These are all the things involved in the aerobic aerobic metabolism. Here next this is the how the organism growth. First they explain the theory, how it is changed, how it is evaluated. After the development, how it is grown in which condition, what is the adaptable condition for their evolutionary growth, that is the aerobic and anaerobic. Then for that hypothesis evolution, this is hypothesis evolution in the valvocales. This is the once unicellular how the unicellular produced unicellular evolution. This is the one concept. In this concept I am going to explain with this small pictorial representation. And this is the one cell, is a chlamydomonas. You all know this 11th standard itself we studied the one cell molecule is a chlamydomonas. This chlamydomonas, 4 chlamydomonas are arranged in order, it's a guanine. Then after the arrangement, that 6 molecules are make a circle. This circle is a pantorrhea. In this period, that it's forming the circle, it's a biggest circle, and so each by that 8 cell, that 6 chlamydomonas having the pantorrhea, then 8 having it's a group, it's called as the Yamagicilia, and that makes some changes in the arrangement, that makes a interaction, it allows the other molecules called as the eukaryotic. Then you can see the maturation period, that is the eukaryotic. After the maturation, that paleodorina, in that you can see that upper part cells are in immature, that smaller ones are in a mature form. Okay, in this maturation they have a 2 type, female, male. The females are round shaped one, and this males are the sticky like one. This is the anisogamy. This maturation is called as the anisogamy. So in this image itself you can see the external parts having immature one, that is the new unicells, and internal part they are producing the female and males, that is the anisogamy. If they those two is types 1, this internal part it's a make a group, and it's this all the things are mature things in the cell. It is having 3 types, isogamy and anisogamy. If there is an isogamy means 2 chlamydomonas, that is a mating type is a this is a mating type plus, this is mating type minus, they are make a mating with their that external flagellate on the structure. Then the anisogamy, and it's the another type in the female they don't having the external flagella, but male having the flagella. So that that male having the mating with this females. This you can understand this concept. I think isogamy means it's make a mating with this they both are, one is plus, one is a minus, that makes interaction with the that flagella that external part. Then it's anisogamy, it's both are having the flagels, and the female it's in a round like form. That another one is the ogamy, it's a female having the round, and it's having the sticky like structure. This another example. Here I will explain the concept. Here the unicellular, then multiple flagellas are arranged and aggregated, and unspecialized flagellate cells from a holosphere, that form it's forming the holosphere. Specialized reproductive cells from cells forms, that reproductive cells are formed. Then cells being in the fold in the make a tissue. This round it's make a fold, then it's closed, and in it make a close and divided into the circle. This external part make a circle. Then evolution of the eukaryotic cell, how it is evaluated. In this ancient time, the aerobic bacteria is entered and nuclear development happened in the the anaerobic bacteria prokaryotic. This is the anaerobic prokaryotic. This is added with the bacteria. Here you can see the mitochondria is there in developed in the cell, and aerobic eukaryotic formation. In that first step, you want to know ancient anaerobic probiotic is here. With that eukaryotic, that prokaryotic cell, the bacteria, aerobic bacteria is inserted. Due to the insertion, there is the aerobic eukaryote that having the mitochondria. Then this is converted into two types. This conversion happens with the if the bacteria are producing the photosynthesis molecule. This primitive photosynthesis eukaryotic are formed, that's and photosynthesis unicellular eukaryotes are produced, that is the plants and photosynthesis unicellular eukaryotes. Another thing is the animals, fungi and unicellular eukaryotics are formed. Now this is the origin of eukaryotic cell evaluation. In this cell, if you can image itself, you can understand aerobic bacteria and anaerobic prokaryotic. It's a very important term. Aerobic bacteria and anaerobic eukaryote. Here the main thing is the conversion of this mitochondrial conversion. Mitochondrions are formed. This mitochondrions, if there is cell having the mitochondrions that producing the animals and plants, that in that another way photosynthesis of eukaryotic, it forms the develop the plants with the unicellular eukaryotic. Okay. Another thing is evolution of prokaryotic. In this prokaryotic, where it's developed in the three types. It's a bacteria, archaea and eukarya. This all things are developed in the evolution of the evolutionary developments, that is the divisions. Okay, then first thing is how it is the first cell originated. In the first cell origination, the first origin of the first cell is a primitive from earth using the cooling. The primitive earth is cooled and produce the inorganic chemicals. These inorganic chemicals are capture the energies from the environment that are producing the small organic. That small molecules are polymerized and the polymerizations happen. That is a plasma membrane and micromolecules, macromolecules are produced. That is a very important step in the evolution, that polymerization, the small inorganic molecules are converted into the plasma membrane. Okay, there is a plasma membranes are there that makes the protocell. There is no internal cell parts arrangements. Okay, so into the soil there is a biological evolution that makes a cell. That cells are converted into the photosynthesis cells and cell respiration process. If that are doing the cell respiration, that's are converted into the animal cells, that is the photosynthesis, that are converted into plant cells. Okay, this is the chemical productions. Or the animals and plants also everything are producing the chemicals. How this is experimentally explained with the Miller-Urey experiment. Who is that? Who is the person means? In 1953, he explained the Stanley Miller and the Harold Urey. They tested a part of the Operon Haldane hypothesis. What is the Operon theory? This is the Operon theory. As per the theory that Haldane showed as
compound that makes a simple molecule. That simple molecule makes a complex building. That complexes are aggregated in a form. That aggregations makes a protocells. That protocells are additionally added with the nucleus. That is that nucleus having protocell. It's called as the cell. It's the Operon theory. It's very important. Who explained the first cell means that's Haldane theory. Is that as per the theory that theory is evaluated by the Miller experiment. Its application means it indicates the the experiment indicated that if the atmosphere was made up of the gases, okay, right, the environment is having the gas that it could have the form the basic organic compound as per their Operon. They said, right, that small molecules are the macromolecules. How the small molecules are produced? That explaining experiment is the Miller experiment. In here you can see the molecule, that boiling chamber. This is another image I will explain. Here there is a water. In the earth, naturally that having the water and the environment. This water is boiled using the heat. This boiling make evaporation that is added with the gases. That environment having gases. That electrodes are making spark. This gases are methane, nitric acids, hydrogen or oxygen. Here they evaporated water. All those things are here, and this that reaction happen in this particular place with the gas. That reactions make after the reaction that make a gas that is a evaporated content. This content is hot water outlet. Here this is the cool water entry. This cool water entry, what did this cool water entry? This cool water contains this extra compounds. So the water droplets, that water steam make an interaction with the gas. That interacted make a reaction. That reactions are condensed and forming the small organic molecule. These small organic molecules are settled down in the tube. Except waters are returned back to the boiling chamber. This is the Miller's experiment. Then here another way. In the theoretical, they mention in the laboratory scale, they mention it in the Miller's experiment. In normal plant and animal, that how it is produced, the abiotic synthesis of macromolecule. Its replication and metabolisms are key properties of the life. In the abiotic condition, the replication and metabolism. That protobiotics and aggregates of abiotically produce the molecule surrounded by the membrane. Without the maintenance of abiotic condition, we can't produce the chemicals. Then it's exhibit this simple reproduction and metabolism and maintain its internal chemical environment. Then basic molecules are some of the basic molecules. This is the 1920 they Operon explained is by Russian chemist, and Haldane, it's English genetics. It's a British person. It's a genetics person. It's independent suggests the origin of life and the evolution of the earth. As per their theory, they reported the how the chemicals are produced. That is the water and environmental gas reaction. Water, heat, gas. These are all these three things involved in the evaluation of the production of biological molecules. The very important thing I want to once again tell you, the water is a major thing. Heat and the gas. These are the things are naturally available in. So due to the reaction of water and heat, that makes steam, that steam and the gas reaction, that gas makes the compounds, and these are the contents after the particular chemical that cooling, the cooling happen in the environmental change, that cooling makes the molecules. Okay, here another this basic biology of origin of basic biology. Here the is here they mention the atmosphere of earth having the water, methane and ammonia. Methane, ammonia and water, these are all naturally present in the earth. These are protein and lipid and nucleic acids. This series the sequence, okay, water, methane, urea, it's converted into the protein, lipid, nucleic acids. These are all called makes the colloidal bodies. This is the co-servate. This is called as explained by the Operon and Haldane. Tomorrow we will discuss the remaining part of 11 and unit 8. That's the second section 2, the last topic. Today is the last session, that session 2's last day of session 2, that is 23rd day. We decided day 11 to day 13 to day 23 is part for it's a timing for the session 2 discussion. Tomorrow I will try to finish the content, and I will also send some notes and a video for the topic, today's topic. Next point, one thing I want to let you know, tomorrow, day after tomorrow onwards, we are going to discuss that session 3, that is somewhat important topics, that is not that as per their previous year question consulting, we are having the some little bit important topics. That topics I will discussed in the session 3, that is the 24th day to day 30, that is 7 days. In that 7 days we will discuss some topics. Okay, thank you for your cooperation and patience. If you have any queries, please post it in a chat box. I will try to answer, or I will make take the questions and ask to their specialist. Okay, Tamil Selvam, AC maker in Lawson molecule. Okay, Ganga Dev ma'am, dominant versus regressive characters, make a one. It is what is SAP effectors? This SAP effectors are helpful for their 1 minute. I'll let you know. Differential effectors having the antibiotic, that is a production of that makes the signals to the production, that multiplications in the it's make a neuronal system, that make a signal that to neuronal it's present in the neuronal system. And it's make a immunity to the cells.